Hey everybody, welcome back to General Biochemistry Lecture. This time we're covering Chapter 3 from Leninger, Principles of Biochemistry. This chapter covers amino acids, peptides, and proteins. And I'm not gonna lie, it's one of my favorite chapters. These are the key principles that you should be able to understand and expand upon by the end of this lecture. So make sure that you go back to this slide and look at these 4 principles, make sure that they make sense. We'll start with talking about amino acids. We're going to do a little bit of a review of isomers because you need to know some terminology. An isomer is a compound that has they have the same chemical formula but different properties. And from there, you can branch off to talk about structural isomers, which have different bonds. We're not talking about those. We're talking about stereoisomers, which have the same bonds, but different spatial arrangements. And within that category, you can have diastereomers, where you're talking about cis versus trans arrangements, or enantiomers. And those are the mirror images. That's what we're going to be talking about today when we talk about amino acids, enantiomers. So just a heads up, if you don't remember that from general chemistry / organic chemistry, you may want to go back and review that. We're not going to be too heavy in labeling things as diastereomers versus enantiomers or anything like that, but you want to make sure that you understand what an enantiomer is and what that means in terms of amino acids. So this is the generic structure for an amino acid. There's an alpha carbon at the center, and that alpha carbon is the chiral center. All amino acids except for glycine have a chiral carbon at the center. The shape is tetrahedral, and they all have this amino group here and this carboxylic acid. There's a hydrogen, and then there's this R group, which is different depending on the side chain. The side chain is what gives each amino acid its own little flair. Okay. So we just went over all of these. The carboxyl group, amino group, hydrogen, and then the R group, which is the side chain. There are actually 2 possible stereoisomers for amino acids, and they are enantiomers, which means that they are a mirror image of each other. So if you were to have a mirror right here, what you would reflect is if you had the L-alanine, let's say, looking in the mirror, what you would see is D-alanine. And this matters because the spatial arrangement means different things in terms of binding. Optically active, that just means that if you had 2 separate mixtures, one of L-alanine, pure L-alanine, the other one of pure D-alanine, let's say, since we have alanine on the slide here, then you would see differences in terms of how those 2 compounds interact with light. So all this means is that these enantiomers interact with light differently. And you can detect that. Not going heavy into that either. I just want you to know that that's what optically active means. When you're looking at amino acids, there's the D and the L. and that tells you what configuration amino acid you're looking at. For our purposes, we're focused on the L amino acids because that's what you see in proteins. There are 5 main classes of amino acids: nonpolar aliphatic, aromatic, polar uncharged, positively charged, negatively charged. You may be used to seeing fewer classes. Sometimes these two groups are lumped together to just say hydrophobic, and these groups are lumped together to just say charged. But we're going to have a little bit more detail in this class. Let's start with the nonpolar aliphatic groups. All right. So, like we said before, all amino acids have the same amino group, carboxyl group, and hydrogen, but what's highlighted in this coral color is the R group, and that's what gives them their specific properties. Aliphatic simply means like a carbon chain. We'll be a little more specific, hydrocarbon chain. Now there's one exception here. There's a sulfur in this methionine, but it's still largely it's an aliphatic group. Okay. So all of these are nonpolar in nature, and the hydrophobic effect that we talked about in chapter 2, these amino acids help stabilize protein structure through that hydrophobic effect. They will cluster together and become stable and sequester themselves away from water. You will need to memorize your amino acids for this class. You'll need to be able to recognize them and draw them. You'll also need to be able to draw short peptides as well. And we'll get to that in a second. The next group is aromatic R groups. So these are the ones that have some kind of, they've got double bonds. You see this benzene structure as a part of all of them. They absorb light somewhere in the 270 to 280 nanometer range. And when you're looking for protein concentrations and you're looking at a particular wavelength of light around 280, you're looking at these amino acids. Now, all proteins don't have a high concentration of phenylalanine, tyrosine, or tryptophan. So if they have kind of reduced numbers of these particular residues, you're not really going to see it that well. That's just if you're in the lab tip. These groups can also contribute to the hydrophobic effect, just like the nonpolar aliphatic groups. Then we've got the polar uncharged R groups. So they can form hydrogen bonds. Cysteine is special because it can form disulfide bonds. So it will make bonds with other cysteine residues and form these disulfide bonds, which you may also see termed disulfide bridges. And those are also very important for protein structure. We've got positively charged R groups. They have a significant positive charge at a pH of 7. And remember from chapter 2, pH 7-ish is pretty biologically relevant. So the charge around this pH is definitely relevant to protein structure and function. Then we've got the negatively charged R groups, and they have a net negative charge at pH 7. Again, you'll need to know these, so make sure that you recognize the structures and that you know the names. There's also some uncommon amino acids that have important functions. You don't need to memorize this. This is just for your own edification. So some common ones are 4-hydroxyproline, and that is a modification that happens after a protein is synthesized and is found in collagen. Then sometimes you have modifications that occur during protein synthesis, such as pyrolysine. Another one, we've got modified transiently to change a protein's function, like phosphorylation. We're going to talk a lot about those, especially when it comes to metabolism. And then we have some free metabolites like ornithine. It's an intermediate in arginine biosynthesis. Sometimes intermediates can act as part of feedback mechanisms to control metabolic processes. So all of these are uncommon amino acid examples that actually have pretty big roles. Again, you don't need to memorize this. This is just for your knowledge. Amino acids can act as acids or bases. So if you're a little bit shaky on chapter 2 with how weak acids and weak bases, we talked about equilibrium and pKa's and things like that, you'll definitely want to shore yourself up on that and then revisit the amino acids. All amino acids have a non-ionic form. I'm going to break up my highlighter here. And in this non-ionic form, that means that there's no charge. So NH2, COOH, no charge there. The Zwitter ion form we've gained a hydrogen here. And we've lost a hydrogen here. The Zwitter ion occurs around neutral pH. And what that means is that this form has a net zero charge. So that's the Zwitter ion. Net zero charge. Zwitter ions can act as an acid or a base. If it's acting as an acid, it's going to lose a hydrogen or donate a hydrogen. So I'm just highlighting here, we go from that NH3 to NH2. So we're donating a hydrogen. Zwitter ions can act as a base by picking up an extra hydrogen. Should write that on the other side. So this acts as a proton acceptor. And we go from a negative charge here to no charge. So Zwitter ions are very important. When we're looking at a titration curve, like the ones we talked about in chapter 2, we can get a lot of information. We can get the pKa of each ionizing group from an amino acid. So each amino acid has at least 2 ionizing groups. The amino group and the carboxyl group. Right now, we're looking at glycine and its titration curve. Its R group is just another hydrogen. Clearly not hydrolyzable, it is connected to a carbon. What this means is that since you have 2 ionizable groups, there are 2 regions of buffering power. So these blue rectangles show the 2 different buffering zones that glycine has because of these two ionizable groups. You'll notice this pi, which we haven't introduced pi yet. Pi is the isoelectric point. The isoelectric point is the point where you have the ion, the Zwitter ion, with the net zero charge. So if we're looking at this and trying to figure out which species of glycine was present, at very low pH, you're going to have this form here. At about 7, so our pi is 5.97, so somewhere around neutral pH, you have that Zwitter ion. And then at high pH, or basic solution, you're going to have this last form. We're going to walk through this for different types of amino acids in class. I'll show you a couple more examples, but understanding a titration curve and which species of amino acid is present is definitely something you'll need to do for this class. We're not finished talking about titration curves, but I want to give you the heads up now. So in general, when you're looking at only two ionizable groups like with glycine, you start with the cation, which is something that's positively charged. Zwitter ion, zero charge. And then anion, which is something that's negatively charged. Make sure that you understand this. We'll talk a little bit more about that isoelectric point. If you don't have an ionizable side chain, remember that R group, then you can calculate the isoelectric point by taking the average of pK1 and pK2. And pK1 and pK2, it's another way of saying pKa1 and pKa2. Same thing. When you have pH that's equal to the pi, the net charge is 0. Your amino acid is not really going to be that soluble in water, and it doesn't migrate in an electric field. It's got no charge. When the pH is greater than the pi, you're going to have a net negative charge. And when the pH is less than the pi, it's going to be a net positive charge. These are definitely things you should know, because I can ask you a question about different amino acids, maybe that don't have ionizable side chains, so the nonpolar ones, and ask you at pH X, what do you expect this amino acid to be? You know, is it positively charged, negatively charged, zero charge? So the pKa's for the carboxyl group and the amino group on an amino acid are different from a carboxyl group on its own and amino groups on their own. The normal pKa for a carboxyl group is about 4.8, but it's lowered in this case because the amino group withdraws electrons from the carboxyl group, and that lowers its pKa. On the flip side, the amino group in amino acids actually has a lower pKa as well because of the carboxyl group. So normally the pKa for an amino group is about 10.6. So because these two groups are in close proximity, they affect each other's pKa. Since amino acids have these ionizable groups, they can act as buffers. We've got two buffering regions. The closer you are to a pKa, the better the buffer you have. If I gave you a titration curve, you should be able to tell me where the buffering regions are and what pH you wouldn't want to have a buffer. So in this case, you would not want to have, if you were trying to make a buffer at, say, 7, it's not going to work too well. Okay? So you should be able to look and see, okay, these are my buffering regions. These are the pHs I'd want to be close to, about 2, and somewhere in the ballpark of 10. So remember those amino acids that have ionizable side chains, like the negatively charged ones, positively charged ones? Well, those side chains have a pKa value as well. So now, instead of just pK1 and pK2, we've got pK1, pK2, pK3. And they can act as buffers as well, and they influence the pI of the amino acid. So if you have an ionizable side chain, you can't just calculate the pI like you would with one that only has 2 ionizable groups. You can still do a titration, but you're going to have 3 ionization steps instead of 2. So here are a couple of examples, and these look a little different than the glycine titration. And you can see that you've got all these different, the different pK. So you can have pK1, pK2, pKr, or 1, 2, 3. So in your book, you'll see pKr. I may use pK1, 2, and 3, but understand it's the same thing. Looking at these structures and being able to tell the net charge is going to be key, because that will tell you whether or not you're looking at one of the pKa's or if you're looking at the pI. In the case of glutamate, your first value is pK1. Then the next value that you get is your pI when you're doing this titration. And knowing the different ionizable states of the amino acid will help you identify which points you're coming across as you're doing your titration. In the case of histidine, the order of events is pK1, pKr, and then you're looking at the pI. That's where our Zwitterion is. So you want to take note of that charge. And again, we're going to do some practice in class to be able to identify these things, net charge, drawing the structures, that kind of thing. So that's amino acids. Now we're going to string them together to make peptides and proteins. Peptides are just chains of amino acids. and they're formed by condensation of two amino acids. So you've got the OH from a carboxyl group on one amino acid and a hydrogen from the amino group on a second amino acid. You lose a water and you form this peptide bond. This is an amide bond. You can also break these bonds through hydrolysis. So that is using water to cut this bond apart. Not exactly as easy to do on the benchtop, but enzymes can do this quite easily. When we're talking about peptides, you can talk about them by the number. So a dipeptide is 2 amino acids. Tri means 3, so that's 3 amino acids. An oligopeptide, it's a short peptide. It's got a few amino acids. Polypeptide means that you've got something bigger, more than 10 kilodaltons. And then proteins have thousands of amino acids, so much bigger than 10 kilodaltons. It's just a little terminology. When it comes to numbering and naming, we're going to start with the amino terminal end. So notice this free amine here. This free amino group. That is the N terminal end. The amino terminal is what that N is short for. That is the beginning of the peptide. Then you have all your other residues, which residue is just another way of saying amino acid. And then you have the carboxyl terminal end. Thought I had a different slide for that. Apparently not. So the carboxy terminal end is here, and that's just that free carboxyl group. It's abbreviated C terminal. When it comes to naming peptides, you can use the full amino acid names. We're not going to do that. Ain't nobody got time for that. But you will need to know the 3-letter code abbreviations and the 1-letter code abbreviations. So when you're memorizing the structures of the amino acids, you'll also need to know their 3-letter code and the 1-letter code. The amino acid composition of proteins is very highly variable. So this chart is just a partial looking at cytochrome C. Cytochrome C is a part of the citric acid cycle, or excuse me, the oxidative phosphorylation. It's all kind of linked in the mitochondria. We're going to talk about that when we talk about metabolism. And you can find cytochrome C across different species. We also have chymotrypsinogen in this table. And we're looking at these two proteins and their composition. You know, how many alanines, how many arginines, how many asparagines, all of that. Wildly different. Highly variable. That's all this slide says. You can estimate the number of amino acid residues, which I'm not really going to hold you responsible for that, but it's just something that if you are interested in doing some kind of research, this is one of those tricks that you can use to get an idea of how many residues a protein has. So if the average molecular weight of an amino acid is approximately 128 daltons, and when you take two amino acids and condense them to form a peptide bond, you're removing a water, which is 18 daltons. So that means that on average, you're going to have about 110 for each amino acid. So if you take the molecular weight of your whole protein, divide it by 110, that'll give you roughly the number of residues in that protein. Again, not going to hold you responsible for that. Now, all proteins are made of amino acids. Some proteins also have chemical groups aside from amino acids. So we've got conjugated proteins, and they have a permanently associated chemical component. Emphasis on permanently. So the part that is the non-amino acid part is called a prosthetic group. So we've got plenty of examples. So there's lipoproteins that have lipids, glycoproteins that have carbohydrates. You see that a lot in the immune system. Phosphoproteins, they have phosphate groups that are attached. Hemoproteins, so hemoglobin, we're going to talk about hemoglobin. It's got this iron porphyrin group, which is like, it's an iron sulfur cluster. So lots of examples. Metalloproteins are very important, and they require a metal for activity. So you see a lot of iron and zinc, magnesium, very, very popular in the cell, magnesium. So lots of examples of these types of proteins. And that prosthetic group is essential for the protein's function. Here's my favorite part, working with proteins. So proteins can be separated and purified, and we can use the different properties of our protein of interest to separate it away from everything else. You can separate based on size, charge, binding properties, and solubility. In general, when you're purifying a protein, the first thing you're going to do is take your protein source, whether it's a tissue or a microbial cell that you've overexpressed your protein of interest in, and you're going to break it open. It's also called lysis. This gives you a crude extract. It's all the proteins from the cell or the tissue in solution, including all the bajillion D proteins that you don't want. The second step is fractionation. And so you're separating proteins based on size or on charge. Lots of different things that you can do there. You can salt out a protein where you can exploit the differences in solubility. Lots of ways that you can fractionate, and we'll talk about that in just a second. Then the third step is dialysis, and that will take away all the salt and other things that you don't want with your pure protein away from your protein of interest. When you're storing proteins and when you're using proteins, high salt, metal contaminants, all sorts of things can interfere with the function of your protein. So column chromatography is something that if you are into proteins or enzymes, you may end up doing a lot of as a graduate student, or if you choose to do research, like an MD PhD program if you're interested in pre-med. So this is definitely something that if you're interested in it, you could spend a lifetime learning about this. So the first step with any column chromatography is you have a buffered solution, which is called your mobile phase, that migrates through some kind of porous solid material, and that's called the solid phase. And that porous solid material it can have different properties that you're going to exploit. So it could be charged, it could have some kind of a ligand that your protein is attracted to and will bind to. So that mobile phase is the buffer that has, it's going to make your solid phase ready to receive your protein. Okay. So the next step is to take a buffered solution that has your protein and migrate that through the solid phase. And hopefully your protein will interact with the resin, or maybe it's the only thing that doesn't interact with the resin. That's, also a possibility. And the properties of your protein are going to affect the migration. As the proteins migrate through the column, you can collect them using a fraction collector. And you can detect using, usually using UV vis, so 280 nanometer light, which we said will detect those aromatic R groups of amino acids. You can use that to detect protein. So different types of chromatography. Ion exchange, very popular. It separates based on the sign and magnitude of the net electric charge of your protein. So remember, proteins are made of amino acids, and those amino acids all have, ionizable components. Some of them have R chains that are ionizable. All of that together gives the protein itself a charge at different pHs. So it may be positively charged, negatively charged, or neutral. And you can calculate that and then exploit that to be able to isolate your protein away from everything else. So pH and the concentration of salt will affect your protein's ability to bind to an ion exchange column. You can use cation exchangers or anion exchangers. Cation exchangers, which is what's pictured here, you're going to have negatively charged resin. And you're going to have positively charged protein that interacts with that resin. With anion exchangers, you've got positively charged resin and negatively charged protein. And again, you can achieve that by changing the pH or the salt concentration. There's also size exclusion. Size exclusion does exactly that. It will separate based on size. You'll also see it called gel filtration. Same thing. The large proteins are going to emerge first from the column and then smaller and smaller and smaller. The large proteins can't really interact with the resin, so they pretty much just go straight through. They kind of take the bypass, the express lane, and go straight through. But the smaller proteins are small enough to interact with the matrix, and so they're interacting with the components of your solid phase, and it takes them time to travel through. So the smaller the protein is, the longer it takes to travel. Large proteins elute first, and then smaller, smaller, smaller. Then you can have affinity chromatography, and this is based on binding affinity. So some proteins interact with a ligand, and that's just a small molecule that it will bind to, and it'll bind really strongly. You can exploit that and have your protein bind to that ligand, and everything else will just keep on going about its business. You can also use protein tags, so you can express proteins with a tag that it wouldn't normally have out in nature, and that tag can be attracted to some kind of ligand on a column. So that is also a great way to purify a protein. You can use HPLC, high performance liquid chromatography, and that uses high pressure pumps to move proteins down the column, and that will improve your resolution as well. So here we're just looking at the different steps in the procedure and how pure your sample is. So in the beginning, you've got this crude cellular extract, and in that extract you have a ton of protein, right? Lots of protein here. But in terms of your specific activity, whatever reaction your protein catalyzes, pretty small, because that activity is diluted over a pretty large volume. As you continue to purify and fractionate, your activity, your specific activity increases, because you're removing more and more contaminants until at the end, your last step, you've got a very small volume, but that small volume is all your protein. So your specific activity, very high. And that's the goal. To have a small volume, lots of specific activity from your protein, which means that you've got a lot of your protein. Now, we can't just blindly do a purification process and say, okay, we're done. There's protein in these tubes. I dialyze them, and I'm ready to go to town. You need to use some diagnostic tools to actually make sure that you have the protein you think you have. One of the tests that you can do is looking at the size of the protein using electrophoresis. This is a technique that you can use to visualize and characterize purified proteins. So you can estimate the number of different proteins in a mixture, which is kind of related to that degree of purity, which is the next point. You can identify the isoelectric point, and you can also approximate the molar weight or molecular weight of your protein. So you're going to take your protein sample and use an electric field to migrate those proteins across a polyacrylamide gel. The proteins migrate based on a charge to mass ratio, and they're visualized using Kumasi blue dye. There are other ways that you can stain, but Kumasi blue is pretty standard and it's the easiest. Notice that in this first lane, there are markers. These are molecular weight markers. And that is just a standard that you can purchase that has specific amounts of proteins with known molecular weights. And you can compare how your protein runs to the molecular marker to get an idea of what your molecular weight is. The next lane has uninduced cells, and we're going to talk more about this in class. But if you're overexpressing a protein in bacteria or some other cells, you can crack open those cells. You can crack open those cells before you start overexpression and take a look at what your protein composition is. And then you can induce, and all of a sudden, you see this band of protein appear. That is very likely your protein of interest. Then you lyse the cells and you have your crude extract. Again, you see this large band here. You do an ammonium sulfate precipitation, anion exchange, and all these other, whatever other things you're going to do to purify and fractionate your protein. And you end up with this beautiful looking band at the end. And hopefully it's what you want. When you're running a protein gel, you are very likely going to be using sodium dodecyl sulfate, or SDS. It is a detergent. It binds to proteins and it partially unfolds them so that you're not running a native gel, but you're running a denatured gel. That means that the proteins are kind of relaxed and open, and you're going to have a similar charge to mass ratio for all your proteins. So that the only difference is, well, this protein is bigger than another protein, so it's going to move more slowly than the smaller proteins. So SDS helps to separate proteins by their molecular weight, because we're not taking into account the differences in the protein size and shape. There are ways that you can run what's called a native gel, and that takes the protein as it is and lets it run through a gel. But oftentimes that's for a specific reason, and you're not usually using that when you're doing protein expression purification. If you want to estimate the molecular weight of a protein, you have to have some kind of molecular weight marker proteins. And that's what's, again, in this first lane. Usually, this is something that you purchase and you receive, some literature along with your purchase that tells you what the proteins are and their size in Daltons. If you plot this, the distance of their migration on the gel, versus the log of their molecular weight, then you will get a standard curve. And you can take a distance measurement of where your protein runs, and then you can kind of have an idea of what the log of that molecular weight is, and then from there you can figure out the molecular weight of your protein. It's not perfect, but it's definitely, it gets you in the ballpark. So unseparated proteins can be detected and quantified based on their function. We were talking about the specific activity. Well, there's a difference between specific activity and activity in general. So if you're purifying an enzyme, you can follow the activity of your enzyme to make sure that you're actually, purifying your enzyme and that activity is increasing over time. So activity is the total enzyme units in a solution. Specific activity takes into account the amount of enzyme activity per milligram of total protein. So that is the number that was in the chart a few slides ago that takes into account, okay, we've got all this protein, we've got this much activity. As you purify, the number of milligrams of protein, total protein, goes down, which means your specific activity should increase over time. So over the course of purification, it should increase. Now we're moving on to the structure of proteins. There's different levels of structure. There's 4 levels. Primary structure, which is just the amino acid sequence. The secondary structure is looking at recurring structural patterns. Alpha helices, beta sheets, those are secondary structure. Tertiary structure, we're talking about the 3D shape of the protein. And quaternary structure is when we're looking at more than one subunit. So if you've got multiple subunits that come together to form some kind of complex, that would be quaternary structure. The amino acid sequence informs the 3D structure, and that structure is important for specific functions. So if you know something about the function of the protein, then you may be able to guess what its structure is if you know about other proteins with similar functions. And the reverse is true as well. If you find some unknown protein and you can figure out its structure, then you may be able to guess what its function is. Most human proteins are polymorphic, and that means that they have variations in their amino acid sequence. But remember, with the amino acids, we have different classes of amino acids, and you may be able to substitute one amino acid for another and still maintain the overall characteristics. So you can have something that's aromatic. Well, we've got a few choices for that. You want something nonpolar and aliphatic. There's a few choices for that. Charged. There's several choices for that. So doing those kinds of swaps, it could be a net zero change in terms of the structure and function of the protein. Because proteins are so important to life, in order to study them, we got to be able to take them apart. And Edmond degradation is a classic method of sequencing amino acids. Other techniques for sequencing amino acids and big polypeptides have been developed based on this method. Traditionally, when you're trying to figure out the sequence of a protein, you've got 2 options. You can label the protein and figure it out. You can break the protein apart, figure it out. If you're going to use labeling, there are lots of different chemicals that will label the amino terminal amino group. So remember, the first amino acid has that free amino group, and that is available to do chemistry. So you can modify with all kinds of different chemicals to label that group. And from there, you can do a whole host of things to isolate one peptide at a time. And when it comes to Edmond degradation, you're going to first label, and that is at higher pH. Then you can cleave that labeled protein or that labeled amino acid at low pH. And taking advantage of the difference in pH means that you can have labeling complete 100%, and then change the pH and have cleavage 100%. And then from there, you can identify the free amino acid and then start again. Label, cleave, identify the free amino acid. And all of these labeling techniques are based on that kind of a premise. You can also study protein structure by breaking bonds. So you can break disulfide bonds and then see what happens. You can denature proteins. We're not really going to focus on that too much though. One great way to study protein structure is using proteases. Proteases are enzymes that catalytically hydrolyze peptide bonds, far more efficient than chemical means. There's a whole host of different proteases. You don't need to know any of them specifically, but you should know that different proteases have different cleavage points. So they recognize certain sequences of amino acids, and they will reliably cleave proteins in those areas in a certain way. And you can take that information and you can say, digest a protein with 2 or 3 or 5 different proteases, and use the overlapping sequences to help figure out the entire protein structure. You can couple that with mass spectrometry. So you can digest a protein and then use mass spectrometry, which helps you to measure molecular mass with really high accuracy. So you can sequence and you can actually look at the entire proteome, which is all the proteins. We're just going to talk about the general steps involved in mass spectrometry. Mass spec is something that you could, again, spend a lifetime on. Very interesting, very complex, a lot of math, believe it or not. These machines are like modern marvels in my mind, and very powerful tool if you're interested in doing any kind of research, especially like metabolic research where you're looking at a lot of different metabolites. Mass spectrometry is something you'll definitely want to learn about. The first step for mass spec is to ionize the analytes in a vacuum. What that means is you're going to take your peptide pieces, those are the analytes, and you're going to give them a charge. Then you're going to introduce those charged molecules to an electric field or potentially a magnetic field. There's lots of different mass spec. There's lots of different ways to ionize and then introduce to a field. There's different types of mass spectrometry, so it really depends. Then those charged molecules move through the field as a function of their mass to charge ratio, or the M over Z. And then after that, you can deduce the mass of your analyte. Again, we're not going to go into detail about how to do all those things. We will talk about combining liquid chromatography, which is what we talked about with all of the affinity columns and the ion exchange and things like that. You can use liquid chromatography along with mass spectrometry. And you can link up different types of mass spectrometry. So you can do what's called LC tandem MS. You can have mixtures of peptides, and that's how we get to kind of looking at the whole proteome. Resolve those peptides that you first use chromatography to kind of separate, and then look at mass spec. And then you can identify proteins and protein abundance. Very cool stuff. Not going to bore you with it, but if you're interested, let me know. Now, we talked a lot about figuring out the sequence of a protein. You can actually make proteins too. So if you wanted to make a small peptide or a small protein, you can do that chemically. We're not going to talk specifically about the method, even though it is shown here on the right. We're just going to talk about a couple of the main points in terms of how you do this. You have one end of your peptide that's attached to resin on a column. And you protect the groups that you don't want to be chemically modified, usually with FMOC. Okay? Don't worry about what that is. Just know that it's a protectant. You can use whatever chemistry you need to add on your next protein, or your next amino acid, and then you block that, and then you continue over and over again until you have made your peptide or your protein. Chemically, it's not exactly the most efficient. Enzymes are much better at this than we are in the lab. But if you need a small peptide or something for what you're doing in the lab, it can be an alternative to purchasing a peptide. There are actually automated machines that will do this for you versus you having to do all of the steps yourself and be your own organic chemist. So that's all we're going to say here about synthesizing your own proteins chemically. So a couple more notes on amino acid sequence. The sequence of a protein can provide biochemical information. We already talked about how it can inform the structure, so the tertiary structure, and the function. But we can also get information about the cellular location. So maybe there's a tag or a sequence at the beginning or the end of the protein that all proteins with that tag are localized to the endoplasmic reticulum, or there's some kind of prosthetic group that we know binds to this particular region, and that prosthetic group maybe localizes it to the cellular membrane. We can also look at evolution. So the really important residues in a protein or an enzyme are typically conserved. Even when you look at different species, you can look at, so these residues have not changed. These other ones are variable, but at these points, we typically see this amino acid residue here. So on the right, what we're looking at is kind of these conservation charts where the size of the letter indicates how well it's conserved. And then N to C, we're talking about the N terminus to the C terminus. So really big letters, conserved. So in B, that D, which is aspartate, highly conserved. However, there's some variation here. So there's some conservation, but not as much. It's variable there. So this is also called a consensus sequence. You're looking at the most common amino acid at each of these positions. When you do that, you can actually help identify the functional segments of new proteins or unknown proteins. And you can establish sequence and structural relationships. So if we know that this particular consensus sequence is associated with this type of enzyme activity, then we can start to establish different families and things of that nature. We can also identify what's called horizontal gene transfer. And that's when a gene or a group of genes is transferred from one organism to another. When you're trying to look at evolution, these are probably not the genes that you want to look at. And like I said, you can use the evolution and look at these consensus sequences to start defining members of a protein family. Here's a little bit of terminology here, and we're not going to focus too strongly on defining families and things of that nature, but I do want you to have an understanding of how it's done and why it's important. So here's some terms here for you. Homologs or homologous proteins, and they're all just members of a protein family. Paralogs are homologs within the same species. So that would be, for example, in humans, we have different homologs. Ortholog would mean a human protein and, say, a cow protein. And they're members of the same protein family, but humans and cows are two different species. So those proteins would be orthologs. You can compare the protein sequences using a database and identify all of the similarities that would classify these proteins as members of the same family. And we'll do a little bit of that in class to look at some examples and do some sleuthing ourselves. So that's it for this one. That was amino acids, peptides, and proteins. Thanks for watching. Make sure that you tune in a live lecture where we're going to do some practice and we're going to expand on some of the more complex topics to give you some hands-on experience with working with them to prepare you for the exams and assignments that will be coming later on. Have a good one. Stay safe.